Okay, this is the first time I've tried this. Hopefully it's going to work out well since I'm unable to be with you today. But this for you is Chapter 9, Genetics, basically. Mendel and how traits are inherited. So Mendel is considered to be the father of genetics. In other words, he was kind of the first person to ever figure out how traits were inherited. He was an Austrian monk, not Australian. Austria as in the country that's in Europe, Central Europe, south of Germany. He's born in the 1800s, and he became a monk, and he did this genetics research actually as a monk in the monastery. For thousands of years, farmers had been crossing certain plants and animals together because they knew they could get the traits that they wanted in the offspring. But they didn't know, how it happened or why it happened. Mendel was the first one to really put the science along with the breeding. Basically, what Mendel did in his experiments, he worked with pea plants, took different traits and crossed them together, and he wanted to see what he'd get in the offspring. And then he would count how many of each thing that he got. A lot of people say, why in the world would he use peas? Well, first of all, he had to have something that was accessible to him. He lived in a monastery in Central Europe, so that didn't mean that just any old organism that he wanted was available to him. But it turns out peas work out really well for genetics because it's easy to see what traits they have, so it's quick to count, things like flower color and things like that. They're also really small plants. They're pretty easy to grow. And probably the most important part is easy to control which plant pollinated another. I think you'd agree. Think about this in your head here real quick. If you're doing genetics experiments, do you think it's important to know who the parents are? The answer to that, of course, is yes. If you want to know how traits are inherited, you need to know who the parents are, what traits they have, and see how those traits are then passed on to the offspring. And Mendel was able to do that with peas. Here are some of the traits that Mendel looked at. He had round or wrinkled seed, yellow or green seed, different flower color, purple or white. Here I wanted to show you kind of what these are not pea flowers. Pea flowers are really small, so I wanted to get a bigger flower so you can see this. But when Mendel was doing his experiments, if he wanted to make sure that a plant was going to cross-pollinate, in other words, get pollen from another plant, he would simply remove the male parts. He cut off the anthers here. See, without anthers. So this flower only has the female part. The only way it's going to be able to reproduce is to get pollen here on the pistil from another plant. Over here, this is a normal flower that has the female part pistil in the middle, and the anthers are on the outside. You see the pollen on there is orange. So this plant can still self-pollinate. And so we just said to prevent cross-pollination, or excuse me, to prevent self-pollination, Mendel removed the male parts. Over here, if he wanted to ensure that this plant self-pollinated and didn't cross-pollinate, in other words, it didn't get pollen from another plant, think about how he could do that real quick. What could he do to make sure that this plant would not get pollen from any other source? And don't make this question too hard. He simply covered them. Took a little piece of cloth, put it over the flowers, tied them on, and that way he was guaranteeing that this plant here would self-pollinate, reproduce with itself, instead of getting pollen from the outside, brought in by an insect, bee, like that, or wind, possibly. Okay, moving on. There's 6 huge vocab words when it comes to basic genetics, and if you understand those, you've really got it down. The first 2 are dominant and recessive, and you're probably already familiar with these. But if there is a trait that is just controlled by one gene, usually there's going to be one dominant trait and one recessive. At least that's the way the ones Mendel did. They work like that. We're finding out in real life, though, that a lot of traits aren't controlled just by one gene. They're controlled by several. And there's also, even if they are controlled by one gene, it's not just a dominant or recessive. But there are traits that are like that, and those are the ones that Mendel first started out with in figuring out how heredity works. The dominant trait is the one that gets expressed, and that's just a fancy way of saying in genetics that it shows up. When a gene shows up, we say it's expressed. The recessive is the one that's not expressed. It's masked by the dominant. If the dominant gene is present with the recessive gene, the dominant gene is going to cover it up. The only way you get a recessive trait to show through is if you have 2 copies of that recessive gene. We're going to find out here in a minute that really we shouldn't be calling them dominant and recessive genes per se. We should be calling them dominant and recessive, another vocab word that we'll look at in just a second. So here's what Mendel did. So in this example, Mendel was dealing with another trait, seed shape. So he took a purebred spherical seed or round seed and crossed it with a purebred wrinkled seed. So that was his P generation. Again, no pun intended, P generation. And he crossed them. So he just took a little paintbrush, took the pollen from one plant and crossed it over to the other plant. So these are his F1s. And this kind of goes back to our question earlier. I asked you if you crossed a purebred purple plant with a purebred white plant, what would you get in the F1s? Well, you almost needed to ask me a question back. And that's, which trait is dominant? In the case of flower color, it was purple. Purple's dominant over white. So all of those F1 offspring would have been purple. So in this case, spherical seeds are dominant over wrinkled. So all of these F1s are going to be spherical. But they kind of have a little secret to them, right? They're spherical, but they also carry a gene for the wrinkled. Even though they carry that gene, it doesn't show up because it's recessive. The recessive gene gets masked by the dominant gene. So as these F1s now, in the final step of Mendel's experiment, are allowed to self-pollinate again, we're going to get the F2s. What do you think we're going to see in the F2s? Are we going to get all spherical, all wrinkled, or a combination of both? The answer to that question is a combination of both. And that's because those F1s carried genes for wrinkled, even though they were all spherical seeds, they carried wrinkled genes, which means that in the F2 generation, and this is the ratio that Mendel always got, 3-to-1 ratio, which means there were 3 individuals with the dominant trait, 3 individuals with the recessive trait. Excuse me, 1 individual with the recessive trait. So that 3-to-1 ratio is what he always counted in the F2s. 3 dominants to 1 recessive. Now, that doesn't mean it was perfect. So let's say he had 100 offspring. You'd expect a perfect 75-25, right? But you got to keep in mind, living things aren't perfect. So sometimes seeds may die, plants die before they, get to be full size and show their traits. Animals may eat some of the seeds or whatever. Some of the seeds mold in the ground. So it's not going to be a perfect 3-to-1 ratio all the time, but maybe, 77-23, 70-30, somewhere in that very close neighborhood, though, of that 3-to-1 ratio. So that brings us to genes. And notice here that I put the word genes in quotes. And that's because Mendel did not know about DNA in the 1800s or about genes. This is all new, relatively new stuff. So I put genes in quotes because Mendel actually called them factors. He said that parents have some factor that they're sending on to their kids to give them their traits. So if you think about it, Mendel's work is even more impressive because he didn't have the benefit of, what we know now about how DNA works and how genes work. He was just strictly going on what he observed and saw in the outcomes of these genetic crosses that he did. So defining what a gene is, technically a gene is a segment of DNA that codes for a certain trait. What you have to remember about genes is you're going to have 2 copies of a gene for every trait. And that's because you have 2 parents. Just like we talked about last chapter with meiosis, we had those homologous pairs of chromosomes, right? Or in a karyotype, you saw the two homologous pairs, or the two chromosomes in a homologous pair. You got one from mom, one from dad. Well, on those chromosomes are indeed genes. So you have 2 genes for every trait, one from mom and one from dad. And that's a little bit of a lie because, like I said earlier, sometimes genes or traits are controlled by more than one gene. But for some traits, there are indeed just one gene that controls it. You have 2 copies of that, one from your mom and one from your dad. Okay, not sure why that's showing up like that if you see a funny screen. But you can see chromosome pairs there, right? Blue representing dad, pink representing mom, and you can kind of see that they both carry genes for certain traits. Here there's 2 tall genes. Here there's a white gene and a brown gene. And you can see that these 4 different plants carry different genes, one from mom, one from dad. Okay, when we write genes, we use letters to represent them. We use an uppercase letter for the dominant gene and a lowercase letter for the recessive. And from this point on, starting right now, we will no longer just call them genes. We're going to call them this word, alleles. This right here is a very important word. Very important word, because a gene is kind of the generic term for the area of the DNA that has the information for that certain trait. But since there's different versions of that gene, we have a special word called alleles. And that's what an allele is. It's a specific version or different variety for a gene. So in the case of height in pea plants, big T is the tall allele. Technically, we shouldn't call it the tall gene. We should call it the tall allele. Little t is the short allele, not the short gene. We want to say that the variations are called alleles. So start using that term. That's the better term to use. So the gene is plant height. The 2 alleles for that are tall and short. So a pea plant could be, what are the combinations? Knowing that you get 2 genes for every trait, what are the possible combinations? Think about what those might be for plant height in a pea. Well, here they are. You could have 2 tall alleles, 2 short alleles, or one of each. And that brings us to our next idea. We said dominant, recessive are the first 2 big vocab words of the big 6. The third one is homozygous. And that is a term that relates back to the word zygote. Remember, a zygote is when the sperm and egg first join together to make that first cell that's going to become the baby. That's where the word zygous part comes from. And then homo meaning 2 of the same. So homozygous is when you have 2 of the same allele. Excuse me. So there's 2 options for that. You could have 2 tall alleles or 2 short alleles. So we would call this one homozygous dominant because it's 2 big letters, or homozygous recessive because it's 2 little letters or 2 little alleles, right? So homozygous is the big word number 3. Big word number 4, heterozygous. Just the opposite. Still referring back to the zygote here. Hetero meaning different. So you have 2 different alleles instead of 2 of the same. So the only way to do that, plant height, is to have a big T and a little t. That's your 2 options. Okay, there's 2 more vocab words that are very important. First one is phenotype. And that's the way you look, the physical appearance of the trait. So when you talk about pea plants, tall, short, purple flowered, white flowered, round seed, wrinkled seed, terminal flowers, axial flowers, any of those traits that you can see is called a phenotype. So think about what some phenotypes for you as a human would be. And I'll give you an example of even a trait, hair color. So what would be some human phenotypes for hair color? So you should be listing things like brown, black, red, blonde, all those kinds of things are phenotypes. Now, one thing to remember, though, about phenotypes, we say pheno-physical. That's the way I remember it. Pheno is the physical appearance. You can't always see every phenotype, though. For example, blood type would be a phenotype. I can't look at you and say, oh, you have A blood or you have O blood. I'd have to do a test to be able to see that, what you are. So, yeah, most things you can just glance at the organism and see. That's their phenotype. But some things are, at the microscopic level, you may have to do a test to see what that is. And the sixth important vocab word is genotype. And that's just what it sounds like. The type of genes you have. What's in your genes, the genotype. So when you ask for a genotype, you're wanting to know these things. What letters, what genes are there? And generally, we don't want to say, if I ask, what's the genotype of this plant? You don't want to say big T, big T. You want to use the proper terminology and say homozygous dominant, homozygous recessive, or heterozygous. So genotype is what kind of genes you have. Phenotype is the way you look. They are two different things. It's very important to note this, that the genotype is what determines the phenotype. And that's just a fancy way of saying the genes you have determine the way you look. And you have known that before you came into this classroom, but now we're making it official and using the right terminology here. The genes you have, the genotype, give you your physical appearance, your phenotype. So applying that to pea plant height, big T or big T little t means you're going to be a tall pea plant. Again, your genotype here of homozygous dominant or heterozygous determines your tall phenotype. Also, you could be a homozygous recessive genotype, which would cause a short phenotype. So the two are very, of course, linked, right? Genotype means the genes, phenotype means the way you look, but the way you look is determined by your genes. Okay, these aren't really pea plants here, but let's assume they are just for the sake of argument. The plant on the left, in your head, what is its phenotype? If you said tall, that's what we're looking for. It's a phenotype. The physical trait is tall. On the right, the phenotype of the plant on the right would be short, the way it looks. Now let's switch and go to genotype. The plant on the left, what is its genotype? Could have been two different answers here, right? It could be homozygous dominant, big T, or it could have been heterozygous, big T, little t. Both would yield a tall phenotype. What about the plant on the right? What is its genotype? Hopefully you said homozygous recessive. Little t. That's the only way you get a recessive trait to show up, is have two copies of the recessive allele. Okay, now we're going to look at tigers. This one's probably kind of made up, but capital S being orange, that's the dominant allele. Little s is white, the recessive allele. So what is the genotype of the tiger on the left? The genotype. It would be homozygous recessive, little s, right? It's the only way you get a recessive trait. What is that tiger's phenotype? You should have said white. That's the way it looks, right? Tiger on the right, what is its phenotype? Orange is its phenotype, the color it looks, and its genotype. What genes give it that orange color? Two options, right? Homozygous dominant, big S, or heterozygous, big S, little s. But get yourself familiar with using those terms when you say genotypes. Don't give me letters. I know you can say little s. I want you to put it together that means that is a homozygous recessive. This is a homozygous dominant, and this would be a heterozygous, two different size letters. And we're going to pick up with the laws of Mendel at a later time. So, as you go through this video, you should be taking notes and writing this stuff down. You can always pause it and restart as you go. You should be able to go through it and take all your notes within about 25 minutes or so. And I will see you then next class.